right, we're going to look at your lab for the onion and cheek cell. You are actually going to make slides that you can see actual real cells. I don't know if you can see me or not. I'm going to turn this a little bit. But if you're at home, you can watch this video instead of actually being at the lab. Hopefully it'll be the same thing. But first I want to show you how to use the microscopes. Here at the middle school we have two kinds of microscopes. This one is called a stereoscopic, stereo because it has two eye holes. You know, you can put your face up to it. This one only has one. There are benefits to both. This is the one, we are not using this one today, but we'll use it in puppy napped. This one has several switches. You have to turn on the main on switch before you can turn on the two lights. Now this one, you don't have to have a slide. You can put your finger under it and look and see it. But it doesn't magnify as much as this one. This one, I could put my finger under there and I could look at it. I could, turn this and focus it in. It's really cool. This one, on the other hand, magnifies a lot better. But you have whatever you put under there has to be see-through. You have to cut it so thin that light passes through it. You don't really have to be able to see what's on the other side of it, but light has to be able to pass through it. So, to use this one, you put it on the lowest setting, which is the shortest little thing. You can turn on the light. You're going to take your slide, and I have this. This is penicillin mold. It's broken, but it'll still work. Anyway, I'm going to put my slide underneath here. I'm going to hold it down with these little feet. I'm going to make sure it's under the light. Then I'm going to look through the hole, and I'm going to move it around until I find something to take a picture of or to look at. I'm going to get it right in the center, and then I'm going to zoom in and focus it in so I can see it. I'm going to make sure it's right in the center. Then I'm going to turn it up to the medium notch. You cannot do this if you put it on high first. Anyway, so I'm going to make sure it's still in the center. I'm going to center it a little bit. I'm going to focus that in. Now I'm ready to turn it to high power. Please do not put it on high power first. When you move it to try to find something on high power, literally a fraction of a millimeter will make it go off your screen. You'll be frustrated. I'm going to turn it to high power. When I do so, I got to make sure that the high power lens isn't going to hit my slide and move it. If it does, I have to start over. So it's not hitting it. Yes, it did hit it. So now I have to go back to the low power, and I'm going to lower it down a little bit, recenter my object, turn it on medium power. I'm going to focus in. I'm going to recenter my object. Then, since I know it's going to hit, I'm going to put this thing down a little bit, then put it on high power and focus it back in so I can see it. So if you're interested in seeing what I did, what I found, that's what you're seeing in the microscope. Move it again. All right, so the first thing, I'm trying to see if you can see, I think you can. First thing we're going to do is our onion cell. I got a little slice of onion. Onion is great for this because it's in layers. So I'm going to scrape on my onion skin, and you see how I'm getting a little piece off. And when you look at this piece, it is so thin that light passes through it. So I'm going to put it on a slide, flat. I don't want it all folded or wadded up. It's got to be flat, because if it's not flat, light won't pass through it. I'm going to put just a teeny tiny drip of iodine. If you put a full drop, that's probably too much. I'm just kind of halfway squeezing the thing and touching it on there. The iodine will stain the cell. The cells are kind of clear and see-through, so you wouldn't be able to see anything. I put this and it's going to stain the living materials, and I put this little tiny piece of glass on it called a cover slip. Now I'm going to take my other slide out. I'm going to put my slide in on low power. I'm going to find the object I want to see, focus in. Then I'm going to turn it to the medium power. I'm going to make sure I'm still centered and seeing what I want to, focus in. And again, when I turn it to high power, I need to make sure I'm not going to hit my slide or my cover slip. Looks like I'm good. And then I'm going to focus in again. Oh, cool. Again, I will show you what I'm seeing. That's what I'm seeing. You can see the cell walls and the nuclei. Okay, let's see if you can still see this. Okay. So I'm done with that one. You would take this and you would rinse it off and lay it on a piece of paper towel in your sink. The next one I'm going to do is an animal cell. That was a plant cell. Obviously, it had cell walls. The animal cell you're going to get from your own cells. You're going to see one of your own cells today. I've given you a Q-tip. You will literally take the Q-tip and rub it on the inside of your cheek. You don't need to dig a hole. Just rub it on the inside of your cheek for a little while. All right, now I'm going to take my clean slide and I'm going to rub this on there. Make sure I get some stuff off of it. I'm going to put my, got to put a little bit of iodine. Now here's the hard part. You got to put the little bit of iodine where you smeared stuff on here. And again, 1 drop is probably too much. I just barely squeezed the dropper and touched it to that. Once I get that done, I'm going to put my cover slip on, and these have to be clean. If the person last didn't leave it clean, then you need to clean it. Turn this to low power again. Put it in my microscope. Find something to see. There we go. Focus in on low. I'm going to put it, I see a nice blob of cells there. I'm going to turn it on medium and focus it in. And again, when I turn it on high, I'm going to make sure it's not going to hit my slide or my cover slip, and it did not. Focus in again. Got a bunch of them. Okay, so I'm going to show you what I'm seeing. That's what I'm seeing. You can see each cell. You can see the nucleus in each cell. Notice there's no cell wall. Those are cells from my skin. Okay, now what you're going to do, you are going to do your own, and those of you at home aren't going to be able to do that, but the idea is you are going to draw a picture of what you saw. Under the cheek cell, you're going to draw a picture of that slide. Notice the cell walls in the nucleus. That's really all you can really see with our microscopes. You're going to draw a picture of that on a piece of paper. Then you're going to take a picture of it with your Chromebook camera. Your lab paper has instructions on how to do that. Then those are animal cells. You're going to draw a picture of what you see. Notice the cell membranes and the nucleus. Draw a picture of what you see. On a piece of paper, take a picture of it with your Chromebook camera, and those 2 pictures you're going to attach or put on your paper. The instructions on how to do that is on the paper. Have a good day.